Well, hey AP, hope you're doing well when you watch this. This is our continuing discussion on Unit 5 genetics. This is part 3 out of 5 parts. One video is combined into part 4 and 5. And so many years ago, I found this quote: there are always two choices, two paths to take. One is easy, its only reward is that it's easy. We've been learning about Mendelian genetics, but there's, of course, life is so complex, right? And so there are so many inheritance patterns, not just dominant and recessive. Okay, so this lecture starts getting into, all right, there's a lot of different inheritance patterns, okay? So phenotypes, they're subject to mutations. There's multiple phenotypic effects, not just two. And then there's a single character may have multiple genes, and then affected by interactions with even other genes and the environment, which is a crazy one that we're going to talk about a lot in this video, but also I'm going to show you some videos in class. Epigenetic is just crazy. It would be nice to try to, for you to do this problem on your own time so we can talk about it in class. A golden horse with a white mane and tail is known as a palomino. For many years, the genetics of this color was a mystery. Suppose you've been hired by a horse breeder who wants to produce a line of true, so there's like your key here, right? True breeding palomino horses when crossed with one another. So therefore, they're always producing palomino horses, okay? The breeder has 12 palomino stallions that are not related to one another. He tells you that every one of the 12 stallions, when bred to palomino mares, has produced some palomino foals, some chestnuts, and a very cream called Cremello. Not Carmelo, Anthony. That's not a good joke. All right, anyways. The breeder wants your advice on how to produce more palomino foals. Okay, so think about, develop a hypothesis about a genetic relationship between these three types of horses. One location is involved. How many alleles do you think is around? Okay, are any dominant? What breeding experiments could you do to test your hypothesis? What crosses? What types of horses would you cross together? Like a chestnut and, and a palomino, the Cremello and a chestnut. What would you do? Do you think it's possible to develop a strain of true breeding palominos? If not, what recommendation would you make to the breeder? Okay, so just start there for right now, and then we'll pick up this one probably in class. Okay, so new alleles arise by mutation. Teenage Mutant Ninja Turtles, back in the 90s, awesome. Sega Genesis game, even better. Actually, no, Nintendo's original NES game, super cool. New alleles arise by mutations, right? Random changes. Okay, they're rare, super rare, especially if they give an advantage, super rare, but random. Okay, wild type, we've been talking about it this year, but wild type is present in most of the population. Okay, a gene with a wild type allele that is present less than 99% of the time is called polymorphic. So one gene may have more than one allele, okay? It may have more than, two alleles, sorry. And so multiple alleles is the term we use to show that there's a lot of different alleles. So this is an example of this is coat color in rabbits. And so you can see here that we have multiple alleles. We have this allele, and then that's one. And then we have another allele, and then another allele, and then another allele. And you can kind of see that the capital C, kind of beats out all the other ones. That leads to dark gray. You can get chinchilla this way. Point restricted, and then albino is lacking any of this superscripts, okay? And so, do a little problem. A point restricted rabbit was made with a chinchilla rabbit. The two offspring were albino and chinchilla. What were the genotypes of the parents? So you gotta work backwards here. Fill out the box first, and then figure out what went here and here. Okay, work on that one. It's a good idea. Incomplete dominance. Some alleles are neither dominant or recessive. So this actually is a blending. You remember the blending theory, hopefully, of inheritance, which Mendel disproved. Well, this kind of is a blending, right? And so red and white, they make pink here, okay? And then if you cross pink and white together, it's half-half. But if you cross two pinks together, two heterozygotes, you get a 1 to 2 to 1 phenotypic ratio. 1 white, 2 pinks, 1 red on paper, right? On paper. And so human examples of incomplete dominance, familial hypercholesterolemia, which is homozygotes develop really fatty deposits in the skin and tendons, heart attacks during childhood, super obviously sad. Heterozygotes may suffer heart attacks during early adulthood, and homozygotes for the normal allele do not have the disorder. This is genetic testing, and oh man, I'm pretty sure I taught a student a year or two ago that had this, that they were like, yeah, my cholesterol is this number, and they shared it with me. I'm like, wow, that's really high. And then she actually said, yeah, I think, I'm pretty sure I have this. And so, obviously watching what she eats and stuff and exercising, but it's kind of crazy. Incomplete dominance, human examples of it. There's also incomplete penetrance, which means the dominant allele may not always lead to the dominant phenotype. So for example, many dominant alleles exhibit varying degrees of penetrance, which means like showing up. Polydactyly is an example. Extra digits, it's dominant, okay? You're like, well, this is a common misconception. The dominant allele is not always the wild type allele, right? Not always. Why? It's all about fitness. The dominant allele may not confer fitness. So back in the past, why do humans have 5 fingers and not 6? Well, 6 must have somehow led to a decrease in fitness level, according to Darwinian, natural selection and the theory of natural selection. And so codominance. So not all individuals who inherit the dominant polydactyly allele will exhibit 6 complete fingers like that guy. So codominance, they both show up. Co-captains, right? This is easily seen as an example in blood type. A and B are codominant. So A beats O right here. So A is dominant to O. But A and B are codominant, which means you have both types of sugars on, like the carbohydrates on the outside of your blood cells. Okay? And O is completely recessive. Okay, so do these problems now. Again, simple monohybrid crosses. Pleiotropy occurs when a single mutant gene affects 2 or more. So a single gene affects 2 or more distinct and unrelated traits. An example of this is Marfan syndrome. Disproportionately long arms, legs, hands, feet. A weakened aorta, which is like the main artery coming off the heart. Poor eyesight. An example of this is Yao Ming. Some basketball players, sometimes in college, there's been sad stories where in college, these basketball players would play and have a great career, and then they would be planning on getting millions of dollars in the NBA, and they would get tested, and a doctor would be concerned possibly about Marfan syndrome, and they would get tested, and they would be found like, oh yeah, you have Marfan syndrome, and they would not be signed by any professional team, of course, due to health risks. Epistasis is another example of a different way of inheritance. Epistasis is phenotypic expression of one gene is influenced by another gene. So one gene is influenced, so it's influenced by another gene. Okay? We could have some fun with a biblical integration here. I just thought about it. But, do you let others influence you? Do you wear a mask depending upon what situation you're in? Oh, that would be fun. But for time, let's keep going. Coat color and Labrador retrievers. Labs, beautiful dogs, right? Look at this. Black is dominant to brown. But pigment deposition is dominant to no pigment. And so here's the deal. This gene E, let's just do like black underline. This gene determines if B is expressed or not. So watch. Right here. Capital B, capital E, black. But look, capital B, 2 little e's, yellow. Because the pigment deposition gene is not being expressed in a dominant way. So it's probably not making a enzyme protein correctly. So capital B, little e. So you have to have a capital E to get black, and you also have the capital E to get brown, but brown is 2 little b's. Okay? So this is, again, epistasis. You can go on, learn, and we'll probably do some of this in class, utah.edu, pigeons, epistasis, pretty fascinating. Kind of look into epistasis if you want to do well on the test and do that. Learning examples, it helps reinforce the content. It's good. Hybrid vigor means that the hybrid offspring is kind of like has a higher fitness level. You can see right here by the corn. This one looks pretty weak. This one looks pretty weak. This one looks super strong and awesome. That's hybrid vigor. It's a complex trait. Multiple genes are usually at work. Genotype and environment often interact to determine phenotype. Environment. So this is all like nature versus nurture. What determines who you are? DNA or your environment? What you surround yourself with? How you live your life? And it's both, but nature and how you live your life and environment has a big play. So point restriction coat in rabbits. These rabbits have a mutant allele for coat color gene. The enzyme encoded by the gene is inactive at temperatures above 35 Celsius. But when it gets super cold, which our extremities do, the fur in this region gets dark to increase absorption of sunlight rays, right? And to warm up the body, right? Especially these extremity regions, okay? And so epigenetics is fascinating. I'm going to show you some stuff in class, of course, because since we're saving time, we get to like kind of talk and work on stuff and watch some videos. But just identical twins, which this is from the Utah website. Just identical twins, because identical twins, right? DNA is exactly the same. They're identical, right? But notice something. And let's just, I'm going to take time to read this because this is fascinating. You need to understand epigenetics. For well over a century, researchers have compared characteristics in twins in an effort to determine the extent, with certain traits are inherited. This is called behavioral genetics. Classic twin studies have identified a number of behavioral traits and diseases that are likely to have a genetic component. Others that are more strongly influenced, look, same DNA, but it's influenced by their environment. Okay? Finding similarities and differences between these sets of twins is the start to determining the degree to which environment and nature play a role in trait of interest. So let's look at this picture right here. 3-year-old twins versus 50-year-old twins, identical twins. These are epigenetic tags, okay? So chromosome 3 pairs in each set of twins are digitally superimposed. One twin's epigenetic tags are dyed red and the others are dyed green. Epigenetics are like there's these tags, methylation, methyl groups, and all sorts of stuff. Like how you live your life, DNA can be tagged in certain ways. We'll talk about it. When red and green overlap, the regions show up as yellow. Notice, start out with the same DNA. Look at how much yellow here, which means they have similar expression patterns in their DNA. Similar tags. But notice, same chromosome, right? Chromosome 3. But yet, look how much yellow. Not much yellow, right? So these epigenetic tags can be placed in different places, which means their DNA is being expressed differently, which leads to phenotypic differences based upon how they live their lives. Nothing is 100% genetically determined. Not even height. You got to eat, right? But these things do have a play. Reading, autism, Alzheimer's, schizophrenia, alcoholism. We talked about that. Bipolar, high blood pressure, diabetes, multiple sclerosis. They all have a play. Notice the white. They all have a play, but it's never 100%. Never. Okay? Polygenic inheritance occurs when a trait is governed by 2 or more genes having different alleles. So 2 or more genes. Okay? So each dominant allele has a quantitative effect, which means like an additive effect. There it is. There's that word. Didn't read ahead. And so examples, skin color, height, eye color. Here is kind of like skin color example, right? Multiple different genes encode for skin color. Lots of dominance lead to darker pigmentation due to melanin and all the other melanin. I'm forgetting what that is called. But melanin production. Recessive ones, lighter skin pigmentation. And so you can try this one out. Be helpful to do. Alleles and genes interact to produce phenotypes. So there's 2 terms here. Penetrance, which means, proportion of individuals with a certain genotype that show expected phenotype. I write BRCA here because this is like the main gene that's involved with like breast cancer. Highly mutated. When you have a highly mutation, your chance for breast cancer goes dramatically up. I actually just heard a sad story about that. Been praying for that family. But breast cancer goes up, but it's, again, not 100%, right? Penetrance, right? Sometimes people have similar mutations and one gets it really early on in life and one gets it later on in life and one maybe never gets it, right? And expressivity, nice, the degree to which genotype is expressed in an individual. Okay, many people, but not all who inherit a mutant gene of BRCA1 develop breast cancer. A woman with a mutant allele may get both breast and ovarian cancer, but another one may only get breast cancer. So that is an example of variable expressivity. How aggressive is it? Think about Henrietta Lacks, right? Cervical cancer probably isn't the same between, different women, even if it's caused by a similar virus. A lot of factors in play, right? Heritability, here's height, 5'0", 6'5". Looks like a bell curve, huh? Heritability varies. If heritability is 65%, the variation due to genetic, 35% is due to environmental effects, stress, home life, eating, vitamins. Heritability estimates apply only to variation with populations, not individuals, of course, and this is a population of human beings. All right, so this is just examples of how it's not just Mendelian genetics. So knowing each one, making a note card about each one, knowing an example. I love learning about examples in AP Biology. They help me understand the concepts when I took it in the past, biology in college and also teaching it. Just examples and these concrete, illustrative examples are helpful, okay? So, hey, great work. Let me draw you a little smiley face. Maybe like not a creepy smiley face. You probably turned it off, but hey, awesome job. Get some spiky hair, some ears. Are they attached or unattached? Genetic jokes. Okay, never mind. Hey, thanks for watching. You are awesome. God bless. Hope you have a good day.